In previous lessons, we used a naval analogy for ions moving in and out of the cell down their concentration gradients. This movement was mediated by leakage channels, or bouncers in our sailor analogy. Remember that in a neuron, we have a lot of channels that let potassium through easily, but fewer channels that let sodium or chloride through. The key driver of the resting potential is that the membrane is semi-permeable. Potassium ions, British sailors in our analogy, are much more numerous on the inside, and they flow easily through the membrane. Sodium ions, the Americans in our analogy, are more abundant outside, but they can't move very easily between the two rooms. But we saw that even a small leakage of sodium ions will eventually unravel the resting potential, since both the potassium and sodium will be able to merrily travel down their concentration gradients while avoiding developing a charge separation, since they can trade the positive charges more or less one for one. This unraveling of the membrane potential is such a big deal that our bodies spend a huge amount of energy every day just transporting those stragglers back to where they came from, maintaining the concentration gradients. But herein lies the key trick of neuronal signaling. What if instead of just letting a trickle of sodium ions in, what if we had a way of transiently opening the floodgates? What part of the membrane could allow us to do this? The simple leakage channels we've seen so far won't cut it. They have a constant permeability and present constant resistance to the flow of ions. They do what they do, and not much else. To continue our bouncer analogy, we need a more dynamic bouncer who can change his behavior based on what's going on around him, allowing easy flow sometimes and restricting the flow other times. Embedded in the membrane, we have a very important set of channels that fit this bill. They're called voltage-gated channels. These channels change in response to their local environment, opening or closing at any given moment depending on the local membrane potential. At a mechanistic level, these channels are complex molecular machines made from proteins. As the voltage across the membrane changes, the push and pull of the charged parts of their structure shifts the conformation of the protein, either opening or closing the pore of the channel. These channels are amazing bits of molecular clockwork, just a handful of nanometers, billionths of a meter across.